Section 7.3, why are some membranes wavy? So cell membranes, are made of a phospholipid bilayer. And so they have hydrophilic heads that are always in touch with the hydrophilic solution. And then the insides are going to be the lipids or the hydrophobic fatty acids that are interacting with each other. And you know that membranes are filled with proteins and that the membrane is a selectively permeable membrane, meaning polar substances won't go through it because of the hydrophobic inside. So here we have three pictures, A, B, and C. And in A, what you can see is a fluorescent staining of the lattice work inside of the cells. And so these cells are filled with microtubules and other structures that will make a lattice inside of the cell. And so what do I mean by lattice? Well, it's sort of like they have all of these crisscrossing structures inside of the cell, and these tubules are made of proteins. And the proteins give, so there's different types of tubules, but they give the cell a shape. And these things can be broken down or built up. They also provide sort of like a railroad track, in a sense, for other proteins to interact with them. And this becomes really important for building membranes and making membranes of different shapes. So cells are packed with tubules. And here's an example of a cell surface that's showing you microvilli and increasing the surface area by making these invaginations. And so if I'm going to highlight, so here you see this is a cell membrane. This is all part of one cell. Okay, so this is all one cell here. Okay, and so what you can see is that by having waves inside of the membrane, you're going to increase the surface area. So you increase the surface area of these cells with increased wave projection, and with basically no change in volume or very little change in volume. So the volume of the cell will stay about the same. So why do we care about increasing the surface area of the cell? So if you think about a cell and its surface area, well, in the book, the author likes to give the example of sticking your hand in water. So if you had to drink using your hand and you would stick it in water, do you think you could absorb more water with your hand open or your hand closed? So intuitively you would say open because there's more surface area. But an open hand has the same volume as a closed hand. And so increasing the surface area would increase your ability to drink water. And there's some really cool Australian desert lizards that absorb water through their capillary networks on their feet. I'll probably show that in class. And so surface area will increase things like absorbing water or exchanging things with the environment with the internal surface. Over here, I drew 4 examples. So on the left side, we have the hand and a fist. This is my beautiful art. And on the right, we have a root. So plant roots, you would imagine, would have highly branched rooting systems to maximize surface area to increase uptake of nutrients and water, versus a stumpy root, which would have a hard time getting the water and nutrients in for the size of the plant, for example. So that's some examples of why surface area to volume ratios matter. And earlier you learned about surface area to volume ratios, and you did some experiments back in section 4 of the book, and so you can revisit that if you want to. But the surface area of a sphere changes by 4 times pi to the squared of the radius. And so here's some examples showing you that. And the volume changes by 4/3, so just a little more than 1 pi r cubed. So if you start to look at these things mathematically, for example, if you have a small sphere and it has a radius of 1 micrometer, then the surface area would be 12.6 micrometers squared, and the volume would be 4.2 micrometers cubed. And so the surface to volume area ratio would be 3. Then if you took a sphere and you just doubled its size, you notice that the surface area goes from 12 to 50, or 13 to 50, and the volume goes from 4 to 33. And so there's a bigger change in the volume than the surface area. And the surface to volume area ratio of this 2 micrometer sphere is only 1.5. And so what happens is you start losing surface area as the cell grows in size, or the sphere grows in size. So cells aren't all like little round marbles. Some of them are squarish, some of them are rectangles, some of them are flat like eggs. And so these are just sort of some ideas of how how the surface area of a cell would change if it were a sphere. But you could also think of it as a cube. And if you had a cube of, let's say, a radius or a length of 1 on each side compared to a length of 5, and you looked at the total surface area, then it has 6 of these. So the total surface area would be height times the width times the number of sides. So that would be 6. And then for 1 of 5, it would be 150. The total volume would be 1 in that little cube versus 125 in the bigger cube. And the surface to volume ratio would be 6, whereas in the bigger cube it would be 1.2 down here. However, if we take that big cube now and we just make it filled with a whole bunch of little cubes, then the volume is basically the same. It's close to being the same. But the total surface area goes up to 750 versus 150. And the total volume doesn't change between these two cubes. Okay, well, it does a little bit because of the membrane volume, but it's negligible. And the surface to volume ratio then goes from 1.2 to 6. So if you needed a cell that needed exchange sites with the environment, then evolution through natural selection would drive a cell to have a lot of these different types of, would have multicellular structures, so that then you can have a lot of exchanges to the surface area, to the outside. So for example, if you had a cell like this, and then you had components inside of it, let's say it's a multicellular organism, all of the cells on the outside would be happy, because when I say happy, you know, I don't mean really happy. Okay, let's make it. Okay, so we're going to have exchanging of goods going in and out of the cell. The cells closest to the surface are going to get most of the goods, and the ones on the inside are not going to get everything they need. Okay, unless it has the right proportion of volume to distance that it needs to travel. And the bigger this cell gets, so if you get bigger and bigger and bigger, you could imagine that then you might have several layers of these. So the outside ones are all getting great exchange, and then the inside ones maybe don't get everything they need, and then the real internal ones might be stressed. So this is setting up a driving force in nature to keep cell size and organism size constrained unless they evolve new features. So in a human body, we have trillions of cells, and they all need oxygen, and they all need to get rid of their carbon dioxide. They all need nutrients, and they need to get rid of their waste. So in a human, we evolved a vasculature to exchange these goods to the cells, and that there's only about 3 to 5 cells away from a vasculature in an animal like a human. Okay, so then you can also think of these modifications happening in different forms, and this is one large cell versus maybe 8 cubed cells versus a cell with microvilli. And so here we have these membranes that are wavy. And that brings us back to the topic. So the evolution of wavy membranes is important because it greatly increases the surface area. Surface, it's a very fat pen I'm writing with. Area to volume. And so you can have very high levels of exchange with the environment. Okay, so let's look at how wavy membranes came to be. So in your book, there's a couple of examples, and let's start over here with figure 7.16. And so on the top left in A, what we see is that there are microtubules inside of the cell made of tubulin protein, and they function more like railroad tracks. And there are motor proteins, as shown in B. So these are your motor proteins. Here is a motor protein. And the motor proteins attach to the cell membrane, to the phospholipid head, or to a protein in the membrane. And then they walk along a microtubule and pull the membrane out. And they can make these long, narrow membrane, like capillary tubes, but they're like membrane rods in a sense. And so you get these membrane tubes. So this would be the membrane tube up here. And then here you see the membrane tubes forming along these microtubules, which are being stretched and pulled. And so this image down here is showing you a time lapse. So it's still frame of a time lapse video made, showing you, like, for example, this particular membrane is being pulled over time. And so you can see it gets stretched out. It's a little bit there along the microtubule, and then it gets pulled along. And there's another example over here, but you'll notice that the pattern, if you compare the patterns, they branch like at 90-degree angles or 45-degree angles or whatever angle that is. And that's just showing you that the proteins can pull them on the microtubules. And because the microtubules are like at all different kinds of patterns, the membrane then can be pulled along and then branch and go this way and go that way. And so you can make all these really crazy shapes with the membranes as they're being pulled or stretched out. And so those are, that's what this whole figure is about. And so you might want to check that out. The other figure in the book is talking about the force it takes to pull a membrane out of a vesicle or something like that. So in A, what we have here, I'll zoom into this. We have a tube. I don't know if you can see it, but there's a tube there, and it's sticking into a vesicle, which is a cell membrane in green here. And you can then pull this membrane, out of that, like that, in this tube here. And if you have a normal vesicle, which seems to be more rigid than the relaxed vesicle down here, then the tube diameter can change depending on the type of membrane that you have. And so in my drawing, it's hard to see. I'll try to draw this a little better. So you can see this membrane is actually really narrow here. So you have a narrow tube versus a thick tube, and we're talking about the radius of the tube. So a normal vesicle, if it's rigid, more rigid, it will make these narrow tubes. And a relaxed cell membrane here, one that's less rigid, will make thicker diameter membrane tubes. So this then is another example of how natural selection might play a role in driving membrane properties, membrane composition, chemical membrane composition. Okay, Then if we look at this graph, this is a pretty cool experiment. And so what they did was they took 3 different vesicles and they were able to pull them out to different diameter tubes. These are membrane tubes. And what they found is that it takes more force. So over here, over here we have force. So it takes almost no force to pull out a membrane from a large diameter, a relaxed vesicle with a large diameter membrane. And it takes more force to pull out a normal or medium-sized diameter membrane vesicle. But it takes more force, a lot of force, to manipulate a rigid vesicle and a very small diameter tube. So remember, the smaller diameter has increased surface area potential. Or I could just say surface area. And down here, the larger diameter has decreased surface area. So if you needed to maximize surface area, you would want a rigid membrane, but you're going to have to figure out a mechanism in order to overcome this force barrier to pull it out. And so that's a very strong selective pressure to drive evolution to make these kinds of membranes that might be rigid so that you could have small diameter tubes in order to maximize your surface area, which is important for maximizing the exchange sites around these membranes. Okay, so if we go down and look at the table in the book, table 7.2, this is just showing you organelles with different lipids and shapes within different systems. So this is looking at rats versus yeast. And this is, again, looking at their membrane systems. And then these things here are all of the different kinds of substances that make up membrane composition. So like sphingomyelin over here, this you'll find in some membranes. So if we look at sphingomyelin, you find it in the rat liver, the plasma membrane of the ER, endoplasmic reticulum, or the liver plasma membrane, so forth and so on, in the skin. But you go into the yeast. Where are we here? We're here. But if you go into the yeast, you see it's all zeros. It doesn't exist in yeast. And so yeast is a fungi, and it's a much different organism than a rat, which is an animal. And so the composition of the lipids vary across the species, giving you variation in the abilities of these organisms to perform different sort of functions. And this results because there's strong selective pressure through natural selection in making lipids rigid or relaxed. And these chemicals, like cholesterol, play a big role in lipid integrity and lipid function. For example, cholesterol can stabilize lipids to a wider range of temperatures, and so they don't fall apart as easily and they also can make the phospholipids a little more easy or fluid under a wide range of temperatures. So there's a lot going on there. That's all you really need to know from the table. So the last figure is looking at membranes or lipids that are rigid and relaxed, and they have different fluorescent dyes on them. And so in A, if we look at A on the top left panel, you see the blue membrane here is made of rigid lipids, and the red one down here is made of relaxed lipids. And they put them together, and when they heat them up and they pinch off, you can see that the rigid lipid membranes, they form smaller spheres than the relaxed one. You get a bigger sphere. And then they also did some other things where they found that you tended to get the relaxed membranes here versus the rigid ones maybe at a pinch site. They would accumulate in that section. Then different sort of curvatures exist between relaxed and rigid membrane structures. So this is showing you that. But it's really evident here in E. So I'm going to zoom into E. So E is really cool because the blue is the rigid membrane, and the rigid tends to make these more straight parts in this situation, and the relaxed are making these curvy parts. And so you can get something that looks like this. These are all abiotic, non-living membrane systems. There's nothing going on here but just manipulating the composition of the lipids and the temperature. And you get now things that look like microvilli, even though there is no DNA or any living components in this membrane system. It's just abiotic.